Good afternoon, everyone. So, in today's class, we will discuss about the next step in expression, that is mostly with regard to that of eukaryotic expression. That is what we will discuss is about RNA splicing. So why the need for RNA splicing is it's because of the presence of introns, and introns are predominantly majority of the eukaryotic genes have introns. So in this lesson we will discuss about some of the things that you have to remember are you have to focus on is about the chemistry of splicing, then also about splicing pathways. There are three that we will have a discussion on, and then there is something very important, alternate splicing. We will see how one gene can give rise to many different types of proteins, and there are some more things such as exon shuffling, and we will discuss a little more more in RNA editing and also mRNA transport. I hope you remember that we are going, since we started the transcription lesson, we are going with this picture for a long time, right? We discussed about replication, organization of the genome, and in the last class, we have discussed about transcription. And now we are discussing the lesson that is, we also during transcription itself, we have learned about some of the things, the processing, post-transcriptional modifications, which include the GCAP, I hope you remember it, and the poly A tail. And we also have briefly discussed that, or at least mentioned that the introns will be removed and exons will be spliced. So this lesson is about that part of the processing, that is splicing. To start with, I think we have to recollect what we have learned in the previous classes. So in terms of RNA polymerases, prokaryotes have one, whereas eukaryotes have three different RNA polymerases, 1, 2, and 3. One is involved majority of the rRNAs and some others. 2 is the one that transcribes majority of the mRNAs, and polymerase 3 is involved in tRNA transcription and some other small RNAs, right? And we also learned a bit about sigma factor. So there is DNA is there is a gene, and for every gene there will be a promoter, and RNA polymerase has to start transcription from that part of the genome, that is the promoter, right? Broken arrow is the 1+1. So we have RNA polymerase, and this RNA polymerase has to interact with the promoter. For that, we need a factor. In prokaryotes, we have something called a sigma factor, and eukaryotes, the same function is made by general transcription factors. GTFs or sigma factor, as the case may be, or the context may be, whether it is prokaryote or eukaryote, they will bind or recognize the promoter sequences, and they will allow the loading of RNA polymerase. I'll just put polymerase 2 here, right? And we also have seen the complexity of the promoters itself. In prokaryotes, we have -10, we have -35, and -10 in E. coli, TAT, AAT. In -35 is TTG, ACA. And in eukaryotes, you have the initiator region, core promoter elements, downstream promoter elements, and so on. It's a little bit complex. And the promoters of Pol1 and Pol2 are a little bit different than promoters of Pol2. And there is also something I described that you have to all remember about the differences between the prokaryotic and eukaryotic mRNAs, right? That's important because we have to, one second. So because that part, when you remember if a point like this, what are the differences between prokaryotic and eukaryotic mRNAs, then you remember a lot of molecular biology on both the sides, prokaryotes and eukaryotes. First thing, as it is produced, prokaryotes are polycistronic. Majority of them are polycistronic, whereas eukaryotic mRNAs are monocistronic. What is a cistron? It is equivalent to that of gene, the name gene. What is a gene again? Either something that encodes a functional RNA or a protein. That is a gene, which you can also say it as polycistronic. What is meant by polycistronic? That means there are multiple genes under one regulatory or one promoter or downstream of one promoter. Okay, and here is the terminator. So when the RNA is produced, it has multiple open reading frames, and each of them will produce a protein upon translation. And that means in E. coli, in bacteria, one RNA can generate three different proteins because it has three different open reading frames. Some of them can have 6 open reading frames. Some of them may have only 2 open reading frames. The point is the mRNA is polycistronic. Then you know what is meant by monocistronic. Monocistronic is simple. It has a promoter, one open reading frame, and a terminator. So it will produce one mRNA, and that mRNA will give rise to one protein. Whether it will give rise to only one protein or it can give rise to multiple different types of proteins, we will discuss in the as we go in this one. Because I'm mentioning this now, because or I'm specifying a point like this or putting a little bit of doubt, is because we will discuss about alternative splicing. In that, one mRNA can give rise to varied proteins. Okay, so right now, what you should remember is polycistronic in prokaryotes and monocistronic in eukaryotes. And there are also other things. The G cap on eukaryotic 5' end of the mRNA in eukaryotes, and also you have a poly A tail in the 3' end of the 3' end of the mRNA. So these are some of the differences that you should remember. And there is processing because the gene in eukaryotic gene, majority of the eukaryotic genes are they have introns. So there is splicing involved, splicing of exons. That is one other difference, if you want to say, between prokaryotes and eukaryotic mRNAs. So this is basically what summarizes this particular lesson, that you have a promoter region. This is the level of DNA. So you have a promoter region, you can put it like this, that would have been the +1. And you have this particular gene that is shown here has 4 exons. And starting of this exon would have the start codon. And the end one will have, here would be the stop codon. Typically, that is what you will think of. So you don't have to think about start codon and stop codon now, because we are not discussing about translation at this point. But these are intervening sequences. In and tron part is taken from the cistron. You know, cistron is John. Cistron is something like the gene. So they have taken this part, and because they are intervening or interrupting the cistron, they call it as intron, like that. So you have to write it continuous: intron, intron 1, intron 2, and intron 3. So that is at the level of DNA. When the pre-mRNA or the mRNA that is produced right after transcription without any changes, if it has been produced, it will be produced as it will have the exons. It is exactly transcribed what is at the level of DNA with all the 4 exons and 3 introns. And you have a 5' leader region, or you can also say it as untranslated region. You would also have the 3' non-coding region, or UTR, untranslated region. And what splicing does is it will bring all the exons together in the same order as they are on the genomic DNA. Exon 1, 2, 3, and 4. Now we have a start codon here, right? And these codons are read continuously. There is no interruption anywhere. The codons are as such. And then you have a stop codon here. So if ribosome was loaded onto it and it has translated this spliced mRNA, or you can also call it as mature mRNA, then it will give rise to one complete protein. Okay, that is the essence of this chapter. But then we'll have to discuss the types and some more details that are present in this chapter. So in today's class, as I mentioned, we will only discuss some brief things, so you can relax about it. Don't try to, you don't have to feel pressured into it. But then in the next classes, when because you have the textbook, I would like you to have a look at it and come by. And I don't know, I might have given one assignment based on this. I hope you will be able to do it properly. Let's look at how important splicing is in eukaryotes. You can think of, based on, you can also think of it, average number of introns per gene. If you look at the lower or smaller, low complex eukaryotes, you do not find as many introns, where some of them rarely have. But you're looking at some of the model organisms that we talk about: Saccharomyces cerevisiae, Drosophila melanogaster, C. elegans, and Arabidopsis thaliana, mouse musculus. And you see that the number of introns per gene are increasing. This is an average. That means some genes may have more number of introns, some gene may have lesser. We have about approximately we have about 25,000 genes in our genome, in human genome. Okay, and then each gene is having an average. On an average, you're having about 7 introns. Okay, and the size of the introns could be huge. And when we were discussing about the organization of genome, we thought we have come across something called as something where there is no proportionality of the genome size and the number of genes. The genome size is very huge, but it's only around point or very small percentage of the genome is actually coding for the gene, and the other is either junk or it's we don't know the function of it. So with increase in complexity of the organisms, you're finding that there is a large increase in the number of introns. That means splicing is a lot more important, because unless you delete these at the level for translation to happen, the open reading frame should be continuous. That is what I was trying to put you here. If a protein has to be produced, the ribosome has to read this open reading frame continuous. If you had an introns space like this, then the ribosome, it might come to a codon here and doesn't know what to do here, unless there could be a stop codon. Ribosome is a machine, molecular machine. It reads in steps of 3, and it has a something like a cookbook, the genetic code, and it will follow the rules of it. It doesn't matter for the ribosome whether a functional protein has formed or not. It is just a machine. You provide amino acids, you provide tRNA and everything else, it will read whatever is there. For a functional protein to form, there is a need to get rid of these introns. And if this is the range of introns that we have, then we will need to generate mature mRNA which have continuous open reading frames so that ribosome can produce a functional protein. Or I have to say it like this, we have to get rid of introns and splice the exons so that a functional protein is produced upon translation. So some of the things for splicing to occur is we should try to identify or recognize something like this intron exon boundaries, right? Assume here is a splice, exon 1, okay? And here is the exon 2, the green one. So this part is called the 5' splice site. You can think of it is the 5' side of the 5' side of the intron. And here is the 3' splice site, or you can think of it as the 3' side of the intron. Okay, so somehow whichever proteins or components are involved, they should be able to recognize these boundaries. The intron exon boundary on which is the 5' splice site, and also the 3' splice site. If one component has to recognize, then there must be some conserved sequences in these intron exon boundaries, right? So typically the sequences are something like this. I don't need you to mug it up, but I need you to just try to understand at least that there is consensus sequence. What is consensus? That means those bases in this position are, say, conserved like this. So if you see on the end, this is the end of the exon 1, you have C or A in this position, and followed by G. And the start of intron, that is the 5' side of the intron, has G, U. We are saying U because we are talking about RNA. This is at mRNA. So G, U, and in the third position of the intron, 5' side of the intron, you have A or G, and then A, G, and U. So these are some of the sequences that are conserved in these boundaries. Similarly, you also have one more point that is called as branch site. We will come to it, what it means. So there is a adenine that is involved, and then we have here, it could be any of the bases. I think Y is for pyrimidines, uracil, consort. This is for purines. R, I think I will get back to you if in case I am wrong. Otherwise, you can consider it as right. P is for purines, and Y is for pyrimidines, right? What could this be then? Purine or pyrimidine? You will say it as CU, CU. N is for any of the nucleotides. It could be any of the 4 bases, uracil, and here it could be A or G. Okay, but one point in this location, within this sequence, you have adenine in almost all the cases. In these types of introns, you would have A, that is conserved. And also you have several other on the 3' side also. Y11, NC, AG, and G. Okay, what would Y11 be? It will be a stretch of 11 pyrimidines. Okay. Yes. So I hope you understood what it is. You understood the point that there are exon intron boundaries, and there is a branch site, and these 3 sites have conserved bases. Why is that required? Because you have, say, there are certain proteins or components that are involved in splicing. You have several thousands of genes, and each gene has about 7 introns. And if they have to bind, then there should be some conserved sequence here and also here. And that if we can identify, people have identified these bases, and that those are the boundary sequences, conserved ones. So the splicing reaction is typically a trans-esterification reaction, a reaction between COH and a hydroxyl group, and eliminating a water molecule, you'll find a band. So this is the general reaction, and you will form something called as a lariat is here. That is something like what a cowboy would have used, a rope to catch hold of the cows and so on. Okay, let's see what is happening. A phosphate P, a G, a P is for the phosphodiester bond that is between them, like this. So the adenine has 2' OH. You remember the difference between ribose and deoxyribose, right? This will be 1, 2, 3, 4, and here will be the 5th one. So deoxyribose has 2' OH. 3' OH is important for all polymerization in DNA or RNA. 2' OH in RNA, in this particular adenine in the branch site, the 2' OH gets activated and makes a nucleophilic attack with the border, the guanine that is present here, the phosphate of this of the guanine that is present at the 5' side of the intron, or 5' splice site. This is called the 5' splice site, but it is only going to attack the guanine, phosphate of the guanine, which is within the intron. And it makes a phosphodiester trans-esterification. Basically, this is a new ester bond. You have a OH here that is left out because this is broken like this. That OH is now going to make a nucleophilic attack on this, on the phosphate of the G here. This is the 3', the total is called as 3' splice site, but you can call this part of it is the 5' side of the second exon that is present. If you consider this as the exon 1 and this as exon 2, then this is where the cut would have happened, or this phosphate included. And I hope you recognize that the light colored green is the intron part. The dark colored one are the exons. So the 3' OH is going to make a nucleophilic attack and gets joined, or esterification through an ester bond is going to bind to the phosphate here. So exon 1 and exon 2 are joined together like this. What is happening to the intron? Intron is breaking up and is released out, which will be further degraded at a later time. And this is called a lariat, that is identical, or it resembles the rope of the cowboy. Okay. And I would want you to look at the chemistry, which is of how these things are look like in the lariat. This is called a 3-way junction. And you see, I would want you to follow from, I hope you recognize what is this adenine. That is the adenine in the branch site. One second, I will try to see if I have, yes. So what I mean to say, 3 point is you have 1 and this way. There are 3 parts to it, right? This junction, and that is what we are looking at here. So this is the conserved adenine in the branch site, and the branch site, the G, this is the 5' end of the intron. You can think of the intron is this way, and it goes this way and comes here, and this is the 3' end of the intron. Okay. So I want you to think of think of the one of the bonds that we have discussed when we were discussing about GCAP. We found in GCAP you have 3 phosphates, something like this, and then you have the mRNA, right? These are 3 phosphates. Say here you would have had adenine or so. And then we were discussing about a bond, something called as 5' 5' bond. And when we were discussing that, I told that we will have other types of linkages as well. 5' 5' linkage. Why is that 5' 5' linkage? This is this phosphate I'm saying that's alpha, this is beta, and this is again alpha. Until here it belongs to the G, guanine, and here it belongs, this part, the alpha and beta belong to the mRNA. These 2 phosphates, alpha and beta, are on the 5' of the adenine, which is linked to the mRNA. Right? I'm talking about the eukaryotic modification of the GCAP modification. And in the G, you have here, all phosphates are always on the 5' side. They are bound to the 5' carbon. So you have this is 5' and this is also a 5', so you call it a 5' 5' linkage. What do you see here? This is straight RNA and the rest of it is also RNA, and this is also fine. What is different is this is the 5' carbon of this guanine, which is supposedly on the 5' side of the intron, but it made a ester linkage with the adenine at the branch point, that is this adenine, and the linkage is here. This carbon, if we can see it, this would be 1, 2, 3, 4, and 5. Where is it linked to? It is linked to the 2'. So that linkage is at the Y junction, you can call it as 2', 5' linkage. In all of DNA, if you look at, the linkage is typically between, this is the, say for example, this is 5', then you have a phosphate and you have a 3'. All linkages are 5' to 3' linkages, right? But in the GCAP, you find 5' to 5' linkage, and during splicing, you would find 2' to 5' linkage. So that is something interesting if you want to understand it. Say, for example, I could ask a question like, describe what is meant by 5' 5' linkage, 2' 5' linkage, and 5' 3' linkage, and discuss the context with examples. If I said that, then you have to discuss this with in the form of chemistry point of view, and also the biologically important one. All polymerization occurs by 5' to 3' linkage. In the GCAP, you would find 5' to 5' linkage. And in splicing, you will find 2' to 5' linkage, or you can actually call 5' to 2' linkage. This is an odd situation because it's the linkage is a little bit out of path, right? Because there is a 5' end binding to the 2' end of adenine here. Okay. I hope you understood this part of it. So the spliceosome machinery has many components. We will see in the later slides. There are more than 150 proteins. There are 5 different types of RNA. It requires a lot of energy to mediate the reactions or the transesterification reactions. And there are, in all of these, they form ribonucleoproteins. Okay. So that means you have the RNA component and a protein component. I hope you remember such a thing before. We spoke about telomerase. Telomerase is also a ribonucleoprotein, meaning that it has two components chemically. One is made up of protein, the other one is made up of RNA. Majority of the spliceosome machinery, that is the components that recognizes the borders of the 5' splice site and 3' splice site, are ribonucleoproteins. That means they are complexes of protein and RNA. So the RNA components are important for that sequence specificity. Remember, I told you that in the 5' splice site and 3' splice site, there is a bit of conserved sequences. How to recognize those conserved sequences? It is supposedly the RNAs that are present actually aid through base pairing. They aid in the recognition of those sequences. And also they are involved in the catalysis of the reactions as well. So we will find small nuclear RNAs. They are U1, U2, U3, U4, like that. And there are about 100 to 300 bases, not base pairs, you should say, because it's single-stranded. So we'll say bases. And they form something called as SNRNPs, small nuclear ribonucleoproteins. Okay. So they are involved in recognition. They bring the different sites together. RNA splice site, say for example, 5' splice site should come to the branch point A. How does it bend and bring these two sites together? That's again done by these components. And also they're involved in catalysis of the reaction. I will summarize in this particular one, and then we will discuss in the next class how these things are involved in more. Let me give you a general idea of what is happening. Then we can discuss these things in more detail in the next class. There are three things that we are going to read as a part of this splicing machinery alone. There are examples, there will be alternate splicing, and also RNA editing that will come up in the later parts of this lesson. So the three things are nuclear. There are three classes basically, and they are called as nuclear pre-mRNA, and there are group 2 introns and group 1 introns. As the same part, we are also, remind me if I don't, we have to study about ribozymes. What are ribozymes? RNA molecules that can act as enzymes. So we will discuss that also because they are highly related. Here it is the word ribozyme. So majority of the eukaryotic genes undergo this pathway, that class, nuclear pre-RNA, and it involves two transesterification reactions. And there is a branch site A, like we discussed in the previous one. And group 2 introns, it is very rare. It is found in some of the cell organelles of eukaryotes and some prokaryotes. Why cell organelles of eukaryotes? Those are the DNA is present in mitochondria and chloroplast. If you read it, at least as a hypothesis, there is still, the debate keeps on going, but otherwise mitochondria and chloroplasts are supposedly arised by which is explained by a theory called as endosymbiotic theory. Based on the sequences, what you can say is the origin of mitochondria and chloroplast is from bacteria. One is an aerobic bacterium and the other one is a photosynthetic bacterium. So when you see group 2 intron type of classes, you will find those in bacteria or prokaryotes, in archaea along with archaea, and also in these rudimentary kind of bacteria, because now they are completely dependent on on the cell, host cell, right? The eukaryote. So they also have this class 2 type of introns. In group 1 introns, they are also rare, and nuclear rRNA genes is some of the cases it may happen, and organelles and prokaryotes. So you can think of group 1 and group 2 mostly happening. I mean, they are rare to start with, but they are mostly related to prokaryotes. Again, prokaryotes can also include cell organelles like the mitochondria and chloroplast. Okay. Yes. So this is the overview and the last slide for today, and then we will have some other discussions. What is happening? So if we get a general idea, then we will know what else to do. And I find some things interesting and something amazing more. If you look at spliceosome, the pre-mRNA spliceosome class or group 2 introns, the mechanism is almost same. It has a branch site A. This one also has, group 2 also has a branch site A. And then you have the three-way chain formation, and the same thing is happening here too in group 2 self-splicing introns. In this one, the end product is a lariat, and so is in this case too, lariat. What is then the difference between pre-mRNA spliceosome and group 2 introns? Is these are self-splicing, meaning that they do not need so many additional things to recognize and splice out. All these reactions can be carried out kind of like they mediate these reactions, and that is why they also discuss about refer them to as enzymes. If you compare it with that of pre-mRNA spliceosome, you will notice that you have a complex or so many different snRNPs are required to mediate each of these steps. Okay, so that is predominantly, I mean, and again, all the nuclear introns, meaning that they are all eukaryotic, they follow through this class, so they fall under this category. Whereas group 2 self-splicing, you'll find it in prokaryotes and maybe other eukaryotic cell organelles. Group 1 is a little bit different. Group 1 is different in the sense that it doesn't have this branch site. You have a branch site in pre-mRNA spliceosome and group 2 self-splicing introns, but not in group 1. Instead, the group 1 introns fold upon itself like something like this. They fold and create a microenvironment where a guanine is present. This guanine as a nucleotide is present. It is a separate entity itself, in the sense that it is not covalently bound to the intron. Okay, if you just say you wash it, this guanine might just go off also. It is, what I intend to mean is, it is held by non-covalent bonds, but it is bound in a small microenvironment, and there is a OH, that is 3' OH of the guanine, that makes the nucleophilic attack on this one. What happens in this case? The adenine is an integral part of the intron here, right? It is covalently bound, or through phosphodiester bonds, it is bound. It is a part of the intron in this case, or also in this case. But here the guanine is not integral part of the intron, but it is non-covalently bound, and the 3' OH makes a break here, and the 3' OH will bind to the 5' side of the next exon. And so exon 1 and exon 2 are now spliced, and the intron is left out. You can see the difference also here. Here you have a lariat formation. So is the case with this one, group 2 also, you'll find the lariat formation. Whereas in group 1 self splicing, you find it different. It's not the same case, right? It has a kind of cap, G cap, something like that. But there is a difference here. Again, I would want you to discuss the linkage. In this case, we have a 5' to 2' linkage, right? The 2' OH of this one here, the 2' OH of adenine makes an attack here, and then there is esterification happening, and you have a 3-way junction like this. That is something like 5' to 2' linkage. Think of what is happening here. You have a 3' OH. First question is that G nucleotide or nucleoside? Right? Always the phosphates are always on the 5' here. And assume this is the G. So this is 3' OH. That 3' OH is making an attack on the 5' splice site. So basically what you will have here is G, and it has its, on the 5' side, it has its phosphates, alpha, beta, gamma, GTP basically. And here you have a 3' OH. To the 3' OH, you have the 5' of the 5' G of the intron. So it is something like 3' to 5' linkage. Okay. Yes. So if there are, I hope you got a general view of what we are going to discuss in the next class. And we will go into the details of what is happening. And so I think the general, what I tried to do is today to give you a general idea about what we're going to learn. So in the next 2 classes or so, or 3 classes, we might finish this chapter, and then we will go on to translation.